Recently, the International Agency for Research on Cancer, division of the World Health Organization, increased their carcinogenity rankings for 4 insecticides, malathion, diazinon, tetrachlorophenphos, and parathion, as well as 1 herbicide, glyphosate. Although these moves were slightly controversial, it becomes necessary to evaluate pathways of potential human exposure. This study concerns only the herbicide, glyphosate. Glyphosate is considered an organophosphorus herbicide, although it certainly does not display the acute human toxicity of the other industrial organophosphate pesticides. Due to its high NOEL, relatively minor initial acute effects, and high efficacy, glyphosate is registered both as an agricultural and residential chemical. You may know Roundup, the most common formulation of residential use glyphosate. Glyphosate is a broad-spectrum post-emergence herbicide. It functions by inhibiting the shikimic acid pathway, preventing the synthesis of essential amino acids. This study focuses on the potential for surface water contamination via glyphosate from both industrial agricultural use and residential use. California is the state with the highest agricultural output and the highest population, therefore will be the area of concern. The first step of the analysis was to determine if there was a relationship between Roundup-ready GMO crops and local surface water concentrations of glyphosate. The glyphosate sampling data was provided by CEDIT in the California Environmental Data Exchange Network. Data came as geocoded sampling results, which I loaded into a new point feature class for easy visualization and geoprocessing. The agricultural data was downloaded from the USDA's crop data layer as a raster satellite image of approximately 100 different types of crop and land cover. I deleted all non-agricultural data, reclassified the nearly 100 crops into possible Roundup Ready and other. Roundup Ready crops include corn, cotton, soy, canola, and alfalfa. The surface water data was the USGS National Hydrography dataset for California. I created a network class with the surface water line shapefile and loaded the sample locations as facilities on a service area. I then ran a surface area analysis 1, 5, and 10 miles along the surface water network. This is an image of the processed agricultural data over the state of California, and then a zoomed-in image showing all 3 data sets. Using the same image, here are the 1, 5, and 10 mile service areas visualized on top of the existing data. The final step in the geoprocessing was to intersect the service areas with the agricultural data so that each sample location would have areas of Roundup Ready and normal crops within its service areas of differing lengths. This is the graph produced when plotting sampling concentration on the x-axis and percent area on the y-axis. The longer the bar, the more Roundup Ready crops within the service area. The shorter the bar, the less. As you can see, the relatively random percent areas by sampling concentration do not indicate any meaningful relationship as a result of my analysis. The second half of my analysis covered the potential relationship between home and residential use of glyphosate with local surface water concentrations. I assume that suburban homes would be more likely to use glyphosate than rural or urban homes. I determined urban, suburban, rural by use of population density. Density was spatially related to the sampling data by use of the U.S. Census Bureau's census tract data set. The census tracts were intersected with the previously created 1, 5, and 10 mile service areas, and any intersected tracts were counted toward the density statistics. This graph shows glyphosate concentration as a function of the population density in tracts intersected by the service area. Similarly to the first analysis, no meaningful relationship was found between residential use of glyphosate and surface water concentrations. Moving forwards, I would like to improve upon my research by involving DEMs and ArcHydro to better capture the upstream-downstream nature of surface water flows. Further, I would like to perform a more detailed statistical analysis of the data to tease out any relationships I might have missed. Lastly, I would like to run the analysis over using each year of the CDL and only the sampling points taken within 1 year of its publication. I like to thank Professor David Sa and Megan Danielson for their assistance on this project. All inquiries can be directed to my email address, anpatton28@gmail.com.